Okay, this is where we stop. So regulation of digestive activities by secretin and cholecystokinin. What I'm going to do, once we're done, I have like a first part. The study guide is going to be pretty big, as you can imagine, because this is the biggest chapter in AP2. Seriously. Because of the metabolism. Okay, so I'm going to post the study guide today and also have the anatomy study guide with just a list of structures that you have to be able to identify. Images that you're going to see are going to be images from here. I'm not going to use any models for this exam, just images from the PowerPoint. Does that make sense? Good. So, secretin and cholecystokinin. If you don't mind, from now on, we're going to call cholecystokinin CCK. So we don't break our tones. So they play a critical role in the regulation of intestinal and, as we saw, gastric activities. So these two inhibit digestion in the stomach. But we are past that. Let's talk about what they do in terms of the intestinal digestion. When chyme enters the duodenum, what does it contain? Acid and partly digested food. Great. Those are two things that we need to remember. Acid stimulates secretin. Fat, specifically fat, I'm talking about fatty acids, monoglycerides, stimulate CCK. We again face the idea of homeostasis, homeostatic regulation. Presence of acid in the duodenum, is that a homeostatic imbalance? Yeah, acid is not supposed to be there. pH in the duodenum should be about 8. When acid enters, what happens to the pH value in the duodenum? It goes down. That makes sense? How can we increase pH in general? What do we need to add chemically? Base. Yes. We need to add base. Secretin stimulates pancreas to release bicarbonate. Sodium bicarbonate, it's like it's baking soda. Okay? That's base. Does that make sense? So secretin stimulates bicarbonate. I have this, oh yeah, there we go. Okay? What do we do with fat? It's a little bit trickier, but it's actually quite simple. What do we do with fat? Well, it's in the duodenum now. Eventually, we will probably store it or use it. But before we do that, what do we need to do with fat? Break it down and absorb. Absorption of the fat is the separate pain in the rear. We need to break it down. What will break down the fat? Huh? Enzymes, lipase, yes. So we need to produce enzymes. CCK will stimulate the release of enzymes from the pancreas. Does that make sense? So these are effects of secretin and CCK on the pancreas. Secretin is responsible for bicarbonate component. CCK is responsible for enzymatic component. Are we good? That's about pancreas. Now there is another organ, actually two more, that are connected to the duodenum. Liver and gallbladder. Liver produces bile. Gallbladder stores it. Cool? Secretin, to some extent, stimulates liver to start producing bile or to increase the bile production. Am I clear? CCK leads to the gallbladder contraction. Now, I'm kind of running a little bit ahead of the train here, but think about this. Let's just, again, draw it dynamically, okay? It's going to be very simple in the form of sort of a flow chart, okay? This is duodenum. Okay? Chyme enters duodenum. This chyme contains what? Acid and food. Well, let's call it like fat. Just for the purposes of this particular regulation, we'll say fat. Okay? Acid. What does it stimulate? Secretin. Fat, what does that stimulate? CCK. This is pancreas. Secretin will act on pancreas and CCK will act on pancreas too. Secretin, what does it stimulate? Bicarbonate. And CCK will stimulate enzymes. We good? Liver and gallbladder. Secretin acts on liver, stimulates bile. CCK acts on the gallbladder, stimulates contraction. What is released from gallbladder? What? It gets a little bit like webby, but first of all, it's all recorded. Then you saw it in dynamic. I hope it helps. Now, here's the tricky part, not tricky part, but first thing that is produced in that is released in response to fats. Not first, but gallbladder contracts first. Like right now, nobody's eating. We have bile in our gallbladders. Those of us who have gallbladders. I hope everybody has their gallbladder. Okay? So we have bile in there. If we eat something fairly fatty, when it hits the intestine, gallbladder will contract and release massive amount of concentrated bile. Does that make sense? In response to fats. Bile contains bile salts. We're going to talk about them. But the point is, bile salts will be absorbed in intestine, will enter the circulation, will go back to the liver and stimulate bile, which will enter the intestine. Some of that bile salts will be absorbed back and stimulate more bile. And as long as there is food in the intestine, this positive feedback will work out. When chyme goes away, when everything is digested, there's no stimuli, so excessive bile will be instead stored in a gallbladder. Does that make sense? So, in terms of pancreas, secretin stimulates bicarbonate, CCK stimulates enzymes. In terms of liver and gallbladder, secretin stimulates bile production, so production, making new. CCK stimulates bile release. Do you see the difference? Kind of jumping very much ahead of a train. People who, so first of all, gallbladder, is that like a critical organ? It's not. People who have their gallbladder removed, do they have any problems? A bit. With what type of food? Fatty, greasy food. They can eat it. They just can't eat like a, I don't know, a massive amount. Okay? They can't just scoop lard. They need to be careful with that. That makes sense? Because they will not be, and we will see why it's so important. Why bile is critical, absolutely critical for digestion of fats. Okay? When you have got the first portion, immediate release is from gallbladder. And then liver catches up because liver produces it. Does that make sense? So like if you don't have gallbladder, can you still eat a good pork chop? Absolutely. Just, maybe not 20-ounce pork chop in 5 minutes, something like that. So you have to be careful with fatty food. It has to be consumed in a small amount or over a long period of time. Does that make sense? Any questions about this stuff? Mechanical digestion. I find mechanical digestion pretty boring. There's peristalsis. Obviously. It's pretty slow. And there's segmentation. The purpose of segmentation, you can see here in this picture in the right bottom corner of this slide. So this is bolus of food, activity of the circular smooth muscles, break it apart, and mixes it with the digestive juices. That's the whole purpose. And food stays in the duodenum for another few hours. So you have 2-3 hours in the stomach, 3-5 in the intestine. The passing time turns out to be not a full day, but fairly close to it. At the time it spends in the jejunum and ileum and then large intestine, it's a while. Any questions? So, liver. Second largest overall organ in the human body, largest in turn. What's the biggest organ in the skin? Yeah, it's the biggest organ. Liver second. Right hypochondriac region, liver and gallbladder pain here. It's fairly indistinguishable. So if it's a gallbladder generated pain, it's going to be right hypochondriac. If it's liver generated pain, it's going to be right hypochondriac. There are many absolutely fascinating parasitic diseases of liver that unfortunately we don't have time to talk about in details. I may mention a couple. Located under the diaphragm, those the male on the left and female on the right, the liver is removed as far as I can see. Yeah, but it should be like right under the right lung, right here. It's supposed to be here. I don't know where the liver is. Okay. It's probably there. Yeah, there it is. That's liver. And that's a gallbladder. So hepatic artery and hepatic vein supply the blood. Here's an interesting detail, especially about hepatic vein. It brings the blood from GI tract. After the chemicals, after the nutrients were absorbed in the intestine, that blood with absorbed nutrients makes its way to the liver. Why? Huh? Not so much filter, but clean stuff up. If we absorb something that isn't that good, liver will take care of it. What else? It's the major metabolic organ. Say If we eat a lot of fructose, that fructose will be converted to glucose. We eat a lot of like lactose, we drink a lot of milk, galactose will be converted to glucose. If we just overconsume glucose, glucose will be converted into fat. Does that make sense? That's all liver responsibility. It is like a metabolic plant of the body. Consists of 4 lobes. I give you a disclaimer. I'm going to tell you what you need to know anatomically, identify. So there are 4 lobes. Right lobe, which is big. Left lobe, which is smaller. The caudati lobe on top. The quadrati lobe, it's the inferior aspect. You can see gallbladder. You need to know the ligamentum teres and falciform ligament. That's it. Am I clear? So like, I can show you this image. You need to identify. I can show you one of these 4 lobes. You need to identify. Okay. It is covered by the visceral peritoneum. There is a small area here called bare area. There's no peritoneum there. It's pretty soft, squishy. I don't know about human, but mouse liver is definitely squishy. Sampling it is the taking the whole liver is a problem, because you take it with pincers and you immediately damage it. The good thing about mouse liver is that it's huge, and you can get a good sample. If you need to do the tissue slice, you still can get a good sample of mouse liver. Wait a minute. Okay, there we go. I don't know what's going on. Okay, so the basic functional unit of the liver is the liver lobule, which is supposed to be somewhere. Endocrine digestive, let me see. That's a representation of the liver lobule, right? So they are roughly hexagonal. You can see the lobule on the image, okay? And in the center of each lobule, the central vein. Here on the model, this is the one. Central vein collects the blood that flows through the lobule. Does that make sense? So hepatic artery and hepatic vein branch. They branch and they form eventually hepatic vein forms portal vein, and portal veins and artery and hepatic arteries form portal venules, portal arterioles. These blood vessels deliver blood to sinusoids. Can you see these blue blood vessels that I'm highlighting right now? Sinusoids. Okay? Does that make sense? These sinusoids are capillaries that are extremely permeable. Very permeable. There is a very active exchange here in that model. Those, the brown ones, represent sinusoids. They are very permeable, which means the walls are more like a sieve. And the blood flow in those capillaries is very, very sluggish. That allows for few things. First, well, most and foremost, exchange. Exchange between the blood in the sinusoids and the cells of the liver, the hepatocytes. You can see hepatocytes here, this light brown cells, forming plates. Does that make sense? The function of hepatocytes is to absorb nutrients that come to the liver from the intestine and metabolize them. Say, convert glucose into fats, convert fructose and galactose into the glucose. They will release nutrients that are necessary. For instance, if now we are talking about the time between the meals, when you don't eat anything, liver will release glucose in the blood. Does that make sense? If you just ate, liver will absorb glucose from the blood. Sinusoids, due to their permeability, can do that. Another important cell that can be found in the walls of sinusoids are so-called reticular endothelial cells, or macrophages. They will be monitoring the content of the blood, pretty much the presence or absence of pathogens. And they will phagocytose pathogens or debris or something that is not supposed to be in the blood. Does that make sense? So these blood vessels, portal arteriole, portal venule, and the third vessel, it's not a blood vessel, called bile duct, this green one, they always go in triplets. And this is called portal triad. Can you see the portal triad? So the function of venule and arteriole is to deliver blood to the liver, while bile duct collects the bile produced by hepatocytes. Here you can see these tiny little vessels in the right upper corner called bile canaliculi. They will collect the bile produced by hepatocytes, drain the bile into the bile ducts. Bile ducts will drain the bile towards the duodenum. Does it make sense? Okay. Now, what happens to the blood? I think bile is kind of boring, liver dumps it into the bile ducts. They merge and merge and merge and merge. Bile ducts become bigger, 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 bigger. And eventually bile gets in the duodenum. That's pretty straightforward. Now, blood flow. That's more interesting. Arteries and veins that, well, come through the intestinal, through the duodenum, absorb nutrients and deliver those nutrients to the liver, to the sinusoids. Sinusoids will bring this blood to the central vein. The central vein will get it to the hepatic vein. Hepatic vein will deliver it to the vena cava. Ever heard about vena cava before? If you haven't, let me show it to you. If you will switch your attention to this model right here, okay? So this is heart, right? This is liver. I can remove the glass, but can you see that big vessel that comes to the heart from the top? There's a big vessel that comes like behind the liver. It comes to the heart. From the bottom. Can you see the blue one? Like in the center, really, really big. This is, so the one from the top is superior vena cava. The one from the bottom is inferior vena cava. So eventually, blood that goes through the lobules, after it enters central vein and hepatic vein, eventually it will be collected in the inferior vena cava. And vena cava, if you would look at this model again, where does it empty? Huh? That's too many details. Organ, heart. And where does heart deliver it eventually? Eventually, the whole body, right? Lungs, it's a separate idea. It's because of the oxygen. But here's the thing. The blood that will leave the liver will have all the proper nutrients. Does that make sense? So liver will make sure that you have the right amount of glucose, right amount of fats, right amount of proteins, and these nutrients will be delivered everywhere. That make sense? Please understand this idea. Okay, that liver actually kind of dumps it all into the venous blood gets into the heart, makes the circle, as Lexi correctly said, makes the circle through the lung, picks up oxygen, and then oxygen and nutrients finally get delivered to the whole system. Good? Awesome. Bile. Multi-component fluid. Did I make a plug for the book called The Emperor of All Maladies? No, it's in decent time. There's a book, like big time, like capital letters, book, called The Emperor of All Maladies. Disclaimer, I have no royalties from that book. I didn't write it. The author doesn't pay me for promotion. If you want to go to medical profession, it's totally worth to read. I got it from, like, there's hardcover, there's like paperback. You probably can get paperback for a few bucks on eBay or used on Amazon. Library will probably not have it right away. When I got it, I couldn't go to sleep. Not because it was so scary, because I couldn't put it down. It's the story of cancer. It's a brilliant book. I'm not kidding. It's one of the best books I think ever written. It's nonfiction. It's written by a scientist and physician from Harvard Medical School. Wonderful language. Very sober, nothing, like plushy, rosy, we all gonna live forever eventually, medicine is awesome. No. Very sober, very real view of modern oncology. He's an oncologist. And why I talk about it now, he mentions that in ancient times, the idea of, human diseases was an idea that, we have humors, fluids, blood, phlegm, bile, something else, remember. So bile was one of them. So pretty much if somebody was really cranky, the bile was up. If somebody was really, really red, you need to poke the person to let the blood out, too much blood. So fairly, simple treatment. Bile has nothing to do with your mood, I suppose. It is necessary to digest fats. Bicarbonate and bile, of course, needed for the neutralization of acid, but the rest is mostly about fats. Bile contains bile salts, which are necessary to emulsify fats. We'll talk about emulsification. Contains lecithin, same purpose. Contains bile pigments. Bile pigments is what gives color to the feces. Bile pigments actually play a very important role. They allow you to get rid of the heme. component of the blood that's actually toxic. Okay? Your red blood cells get destroyed. You need to get rid of the heme. Heme is broken down into bilirubin, and bilirubin is excreted with feces. Does that make sense? Cholesterol. You have to understand. So cholesterol, what is the main function of cholesterol? Well, not main, but one of the functions of cholesterol in the human body as it is applied to endocrine system. It is the source for which hormones? Steroid hormones, which are produced by two organs. Adrenal glands and like the lowest. Yes, gonads, testes in male, ovaries in female. Does that make sense? These hormones, they, we talked about them, they release practically constantly. So they have to be excreted. That's the function of liver, to excrete steroid hormones. And cholesterol, but essentially steroid hormones. It also produces mucin. As we learned before, mucin is kind of sticky. Okay, so it helps to stick food together. Does that make sense? Awesome. Let's talk about bile salts. This is the bile salt. You need to know the formula. No, just kidding. The idea, if you would look at this molecule, this part is very, very fatty. It actually looks a lot like cholesterol, really. It's a fatty part. It's lipid soluble. The one that has this huge black circle. Does that make sense? This part is water soluble, carboxyl group. So essentially, bile salts are what is called amphiphilic. Which another amphiphilic molecule you know? I guarantee you know it. Heard about it a million times. Molecule that has hydrophobic and hydrophilic ends. Phospholipids, right. Well, bless you, lecithin is phospholipid, not a component of the bile. This is amphiphilic molecule. So hydrophobic tail, which on this representation shown in green, so that's hydrophobic tail, it will interact with lipids. Okay, hydrophilic part will stick out in the intestine. Let me ask you a question. In the intestine, the environment, is it watery or not, normally? Watery, yeah, there's a lot of water. So did you ever try to mix fat and water, like oil and water? Do they mix? Well, no, they don't. Okay? When you wash greasy skillet, what do you use? Dish soap. Why? Why can't you do it with just water? Huh? Why it doesn't clean? The reason is, think about this. If you have a bunch of salt on the surface of the skillet, can you clean it with water? Salt, just sodium chloride, yeah. It will dissolve in water. That's it. You don't even need to use the dish soap. Fat is not dissolved in water. So even if you mechanically, like if you just cook bacon, there's going to be like this thick of a layer of a grease. Yeah, you can scrape it off. It's still going to be fatty. You won't be able to dissolve that thin layer. So if you add dish soap will solubilize the fat. Fat molecules will be in the center, surrounded by the molecules of dish soap. Bile works just as well. Okay, bile will solubilize the fat. It will allow fat to normally float in the watery environment of the intestine. That makes sense. Lecithin works the same way. This structure is here. They're called micelles. Okay, so micelle has a fatty core, sort of, and hydrophilic outside portion. That makes sense. Next time when you eat chocolate, read the content, the ingredients. No, don't worry, it's not going to have bile in it. No. What it's probably going to have is lecithin. And no, it's not human-derived. It's plant-derived. Okay? Lecithin is used in making chocolate. The cocoa beans contain a lot of cocoa fat. It's actually like good chocolate has a lot of fat. And if you don't solubilize it, you're practically going to have like oil clumps inside of the bar. To avoid this, lecithin is added, so those clumps are pretty much indistinguishable from the rest of the chocolate bar. It is emulsifier, emulsifies the fat. Does that make sense? So the function of lecithin and bile salts is to emulsify the fat, break down large clumps of fat into smaller chunks that are digestible by the enzymes. Right? Bilirubin. Red blood cells, and they break down, normally break down after a couple of months in the spleen, by the way. The products of their breakdown reach the liver. Heme, in the meantime, is degraded to bilirubin. And here comes the question. What is the system that is normally getting rid of chemicals, unnecessary chemicals in the blood, normally? What's the organ system that does it? Renal, yeah, it's kidneys, right? Like, you have excessive urea, it will be filtered out in the kidneys. You have, I don't know, excessive potassium, it will be filtered out in kidneys and whatnot. Got it? So what's the problem with bilirubin? It turns out bilirubin binds to albumin. And since it's bound to the protein, the kidneys cannot, they don't let protein to pass in the urine, normally. Healthy kidneys do not let protein to pass through, albumin to pass through. So they cannot filter out bilirubin. Essentially, liver degrades albumin with the bound bilirubin, okay, and excretes bilirubin into the intestine. That makes sense? So bilirubin is what is called bile pigment. It gives, question, Mary, Jessica. Say again? In the kidneys, absolutely not. But liver can digest, pretty much destroy it with bilirubin. Does that make sense? In the renal system, it cannot leave blood. It cannot cross the epithelium of the capillaries in the glomeruli, in the kidney. In the liver, sinusoids are very, very permeable. So there is an exchange even in the protein. Liver actually can produce albumin, which will enter the bloodstream. And it can go back. Does that make sense? Huh? So albumin, if albumin is in the urine, if it crosses somebody's glomerular epithelium, this person is in big trouble. In the liver, it's absolutely normal. Proteins go back and forth. Does it make better sense? So bilirubin ends up in the intestines. Some of it will be excreted with feces. Some of it will be converted by the bacteria in the large intestine in the urobilinogen. Okay? Now, urobilinogen, I probably will have to go one forward. Yeah, so urobilinogen, imagine there's urobilinogen here. It will be absorbed in the intestine. It will go back into the portal vein. It will get back into the liver, get back in the circulation. So essentially, urobilinogen will partially enter the circulation. Now, urobilinogen doesn't get attached to the albumin. It can be filtered out by the kidneys, and that is what gives urine the color. That makes sense. That's why it's amber. Good? Please understand understand why bilirubin cannot be excreted by kidneys. Why we have to involve the liver in it. Does that make sense? Because bilirubin is attached to the proteins, to the albumin. That makes sense? Now, so what is excreted? So I don't really care about the drugs. You don't have to memorize that, say, ampicillin is excreted and penicillin is not. Okay? It's not the purpose. What I want you to understand is that excretion of endogenous chemicals, bile salts, are they excreted? Yes, they are excreted by the liver into the duodenum, but then they can be taken up into the enterohepatic circulation, which means bile salts will enter the blood, go back to the liver, will stimulate more bile salts, which will enter the bile, get delivered to the duodenum, will be reuptaken, okay, and will be circling like that for a very long period of time. That makes sense. Same is true for urobilinogen and cholesterol. Okay? Bilirubin isn't reuptaken, lecithin to a small extent. Okay? Now, functions. I want to be very clear in what I expect you to know about the liver function. First, detoxification. Kupffer cells or macrophages, it's all the same. It's the synonym. Macrophages. Phagocytose pathogens. Hormones and drugs can be chemically altered, excreted, or neutralized. For instance, acetaminophen, which is the acting component of Tylenol, is a drug that is metabolized in liver. That is why it's toxic to the liver. Okay? Now, to give it a toxicity, unless you drink pretty much a bottle of Tylenol, you can hardly experience liver toxicity. It is cumulative. So if you abuse Tylenol on the regular basis, eventually you will kill the liver. Different protein metabolites, urea, uric acid, okay? Liver metabolizes proteins and excretes urea and uric acid either in the duodenum, or those guys get into the blood and then they filtered out by kidneys. Metabolic functions. Conversion of carbohydrates. Okay. Glucose to glycogen and fat. Okay. Gluconeogenesis. So liver can either conserve glucose, conserve energy after the meal. Does that make sense? Or it can use fats and proteins as the precursors for glucose synthesis during gluconeogenesis between the meals or during fasting. That makes sense? Lipid metabolism. Liver produces fat, triglycerides. Liver produces cholesterol. Liver excretes cholesterol. And during extreme fasting or extremely low carbohydrate diet, liver produces ketone bodies. Fatty acids, free fatty acids, are metabolized in liver into ketone bodies. We'll talk about ketosis and dietary ketosis and, weight loss and different types of diet a little bit. Okay, so lipid metabolism. Liver is extremely important for protein synthesis. It synthesizes albumin, which is the essential component of the blood, major osmotic component of the blood, synthesizes beta globulins, which are transport proteins, fatty molecules, lipid-soluble molecules, cannot be transported in the blood by themselves. Why? Plasma consists mostly of water. Fat cannot be transported by water. Clotting factors. Coagulation proteins. Does that make sense? And yes, bile is the liver production. I'm not going to ask about specific pathways just yet, but if I ask you what is the function of liver, you have to recognize clotting factors, albumin production, glucose conversion, back and forth, okay? Storing energy from glucose and making glucose from other sources, okay? Now, a few words about the liver disease. Here you can see the canine liver, top left, that developed cirrhosis. Cirrhosis is the result of inflammation of the liver. Inflammation is a process in which injury is repaired. And it is repaired by what type of tissue? Connective tissue, scar tissue. Connective tissue, can it perform the functions of the liver? It cannot. So, liver function declines. Does that make sense? What can cause cirrhosis? Alcohol abuse. What else? Drug abuse. What else? Hepatitis B and C and E, pretty much, except for A. A is not. A is acute. B, C, D, E can all cause cirrhosis in the long-term perspective. What else? No, thank God. Not cirrhosis. A lot of other bad things, but not cirrhosis. Surprisingly, no. A lot of carbohydrates. When people abuse carbohydrates, they have sweet tooth, like I do. Excessive glucose is converted into fat. Fat largely accumulates in the liver, causes non-alcoholic fatty liver disease, NAFLD, okay, and that leads to cirrhosis. Cirrhosis in the long-term perspective leads to liver cancer, which is practically untreatable. Now, few things. Liver, how long can a person live without a liver? Okay, without kidneys, how long? Well, a couple of weeks. It's going to be miserable life, but a couple of weeks without liver. 18 to 24 hours. That's how important it is. Now, one interesting feature, think about this. If liver is dysfunctional, there's going to be some very noticeable problems in a patient with cirrhosis. Clotting problems, a lot of them. And another interesting problem, not so much cholesterol imbalance, but liver is responsible for the excretion of cholesterol-based hormones. And in males, liver regulates the amount of estrogen. Pretty much it excretes estrogen. Dysfunctional liver will not be able to excrete estrogen, which is normally produced in males. It's a result of testosterone breakdown. Estrogen levels in people with cirrhosis will rise. And one of the symptoms of cirrhosis in patients with, like, let us say, not terminal cirrhosis, but pretty much developed, very well developed cirrhosis, is gynecomastia. As one of my former students who worked in the hospital brought it up when we discussed it, and I explained how estrogen levels increase in people with cirrhosis, she said, "Whoa, now I understand why these patients grow boobs." That's essentially what happens. Okay? Another cohort of patients that have problems with liver, bodybuilders. If they abuse testosterone, if they don't take steroids smart and just shoot themselves with testosterone, that will kill a liver too. Because of it trying to get rid of that massive amounts of steroid hormones. Okay?